Okay, all right. Let's continue to look at the productions of enzymes. Wait, wait. Okay. The productions of enzymes. Okay, extracellular enzymes. So let's look at this. First, you need to know how to interpret this diagram. Okay, so this is the nucleus, and next to the nucleus is the ER. Okay, you still remember what is ER? Endoplasmic reticulum, where you have the rough and also the smooth endoplasmic reticulum. Okay, and you have the blue dots over here. So these, a lot of blue dots here are on the rough endoplasmic reticulum, and it is called ribosome. Okay, so the first one, ribosome, is the site, that means the place of protein synthesis. Okay, so now let's recall, we are looking at how are these extracellular enzymes. Extra, the one that will be used outside of the cell. So we're talking about enzymes. So now enzymes is made up of protein, okay, mostly protein. So that's why it is that's why we need to involve ribosome here, the very first substructure. Okay, so number two, what happened is that this protein synthesized by the ribosome, it will end up entering the lumen of the rough ER. Okay, and you know that this ribosome is on the rough ER, right? So that's why it is called raft. So after being produced, it will enter into the lumen of the rough endoplasmic reticulum, and this is how it is being transported through it. So next what happened is that once it reached the end of the rough endoplasmic reticulum, so let's say this is already the end. So it will form a bud, okay, to depart, buds off to form a transport vesicle over here. Okay, independent transport vesicle over here. Okay, carrying the protein which has been synthesized by the at the ribosome. So next, it is moving on to the next destination. Number 4 over here, contains protein that moves towards the Golgi apparatus and then fuse with it. So next, it is heading to this Golgi apparatus. It is going to fuse with it, and next, what will happen is that, now you see here, in the Golgi apparatus, these proteins are being modified. So again, when you look at this part, you have to go back to chapter 2. You have to study and recall the functions of these cell structures. What is the function of the rough ER, of the ribosome, of the Golgi apparatus? So you know, Golgi apparatus is like an industry, right, where it packages, it modifies, then it secretes. So these proteins are now modified into enzymes, and they are secreted in the secretory vesicle over here that are formed from the tip of the Golgi apparatus. So again, it buds off, okay, and it is now moving towards the plasma membrane. So remember just now we said these are all inside the cell. You see the nucleus is here. So now from here, the secretory vesicle is bringing all the content in it, and it is moving outside. Okay, so once it fills with the plasma membrane, can you imagine, it fills and all the content is going to be released out of this cell. So that's why we say extracellular enzymes. So now these extracellular enzymes, now these are already enzymes. Because now here are still protein. They here undergo modification already. So now these enzymes are ready to serve to catalyze biochemical reactions outside of this cell. Okay, outside of this cell. So that's why we call extracellular enzyme.